Granule cells are these small neurons that you see. They're very compact. You can see lots and lots of punctate or dots, which indicates that these are all probably spines, so they're receiving lots and lots of excitatory inputs. Chandelier cells, I think I already talked about those. Those are those, when it wants to turn you off, it turns you off. It has these capsules of synapses. Here is a basket cell. These are neurons which have tremendously large axon. Here's the dendrites on it, and they synapse to like almost 30% of the excitatory cells within. This is one of the things we'll see. The number of connections between excitatory cells is pretty small. The number of connections between inhibitories can be quite large. These are really, really what connect everybody together. Long distances, things are connected generally through excitatory, but locally, almost all the interactions are excitatory or inhibitory. Here's a retinal cell. This is a ganglion, retinal ganglion. It goes out and receives inputs from lots of areas, and actually is what was responsible for some of the shaping of the connections between these, or what allows certain cells in your visual cortex to respond to certain shapes. So we'll talk about receptive fields. Glia, these are the other cells. It comes from the term glue. I made an analogy which I like, which is these are like the wife cells, in that they are extremely important and underappreciated for a very long time, and now we're starting to recognize just how important they are. And so, everybody sort of said, well, they're just there sort of giving a jelly to support the cells, and then whenever neurotransmitters are released that need to be disposed of, or ion channel balances get out of balance, the glia go and fix it. And now we're recognizing they really do much, much more than that. In fact, there's a whole sort of tripartite synapse, which is you have the presynaptic cell, you have the postsynaptic cell, and then you have glial cells encapsulating it. And in fact, it's so important that you can actually change the calcium inside just the glia cells. So they now have things which you shine a light on and that releases calcium in particular neurons. And you can actually change the strength of the synapse by changing the amount of calcium inside of these guys. So it's not only the plasticity is dependent on the presynaptic, the postsynaptic cell, but the glia controls plasticity as well. Glia are responsible for myelination. So you can see here are axons. You can see all the microtubules going down the axons and an occasional mitochondria sitting in it. And then you have this many, many wrapping layers of mitochondria. So it turns out it's very expensive to maintain all of these sodium channels, voltage-gated sodium channels, to propagate these action potentials. And so what the body has found is an efficient way to do this is to insulate big sections of the axon and then pack the voltage-gated sodium channels just at certain intervals. And what happens is this guy will fire an action potential. The voltage will travel down inside this tube, this insulated tube. The voltage will be just high enough to elicit a voltage-gated response out of these guys. They will start the next action potential and relay it, and it will continue on. And so if you have a disease that actually causes demyelination, you will see the velocities of these dramatically decrease, and the ability in sensory and motor will decrease a lot. So a lot of the tests you'll see, if they're trying to figure out if you have carpal tunnels or you have degenerative myelin disease, is to measure how quickly an action potential will travel down. And they'll put in needles into your arm to stimulate and to measure the response. And it's painful, and so that's the node is called the node of Ranvier, which is packed. The axons, the myelin, they're different cells inside and outside. The dendroglial cells are in the central nervous system, and the peripheral nervous system is a Schwann cell. One of the big differences is I think oligodendroglial cells can actually create myelin on a bunch of cells, and Schwann cells only myelinate one cell, one axon. And the action potential starting at one node of Ranvier and then jumping to the next one and then to the next one, that's called saltatory for a jumping action potential. Here's just different pictures of, this is pictures from Cajal showing the glial cells and the neurons, and here's an example of presynaptic, or one of these is, this is the presynaptic because you can see all the synaptic vesicles. This is the thickening of all of those neurotransmitter receptors, and then here is the astrocyte, the glial cell, which encapsulates it.